Welcome to this video on the required practical, photosynthesis, a GCSE Biology revision focusing on measuring the rate of photosynthesis using pondweed. The big idea of this practical is to use pondweed to investigate how light intensity affects the rate of photosynthesis. Oxygen bubbles released by the pondweed provide evidence that photosynthesis is occurring. Photosynthesis produces oxygen as a byproduct. Therefore, counting the number of oxygen bubbles released by the pondweed provides a quantifiable way to measure the rate of photosynthesis. The equation for photosynthesis involves carbon dioxide and water as reactants, plus light energy absorbed by chlorophyll, yielding glucose and oxygen as products. The essential equipment includes pondweed, like Ilodia, which releases visible oxygen bubbles, a lamp for a controllable light source, a beaker of water to submerge the pondweed and keep the temperature stable, and a stopwatch to count the bubbles per minute. To set up the experiment, place the pondweed, position the lamp at a set distance, allow time for acclimatization, and then count the bubbles. Make sure to let the pondweed acclimatize before recording results at each distance. Light intensity is critical. Moving the lamp closer increases light intensity, leading to more photosynthesis, and thus, more oxygen bubbles. The relationship between light intensity and distance is an inverse square relationship. Shorter distance means stronger light and faster photosynthesis. That's why distance is your independent variable. Knowing your variables is essential for success in this practical and on exams. The independent variable is the distance of the lamp from the pondweed, what you change. The dependent variable is the number of bubbles produced per minute, what you measure. Control variables are everything else kept constant, like temperature and CO2 supply. Control variables must be kept constant so that only light intensity affects your results. This is crucial for a fair test. To maintain constant temperature, use a water bath or large beaker as a heat shield, since enzymes are temperature sensitive. To maintain CO2, add sodium hydrogen carbonate. Finally, use the same piece of pondweed for all readings for a fair test. The method involves setting the lamp distance, counting the bubbles for a set time, then recording the result and moving the lamp to the next distance. You should repeat this process for distances like 10 centimeters to 50 centimeters. Repeat your results at least 3 times at each distance and calculate a mean. Repeating results reduces the effect of anomalous results, improves data reliability, and allows you to identify outliers. When recording results, organize your table with columns for distance, your 3 repeats, and the calculated mean number of bubbles produced per minute. This example data shows how the number of bubbles decreases as distance increases. Plotting your results reveals an inverse relationship. As distance increases, light intensity drops, and the bubble count decreases. This shows that light intensity limits photosynthesis. Now, let's explore limiting factors. What stops photosynthesis from going faster? A limiting factor is a component in short supply that restricts the rate of photosynthesis, even if other conditions are ideal. The 3 key limiting factors are light intensity, carbon dioxide concentration, and temperature. Each one affects the rate of photosynthesis in a specific way. On a graph, limiting factors cause the curve to flatten out into a plateau. This indicates that another factor, like CO2 or temperature, has become the new limit. Safety is important. Be mindful of the hot lamp, keep water away from electricity, and use a heat shield, like a water-filled beaker, to absorb heat and prevent the experiment's temperature from changing. The heat shield, a beaker of water placed between the lamp and pondweed, acts as a filter. It absorbs infrared radiation, ensuring temperature remains a constant control variable. Let's review some key exam techniques so you know exactly what the examiner is looking for. Remember that more bubbles equals a faster rate of photosynthesis. Name your variables correctly. Independent is distance, dependent is bubbles per minute. Always explain why control variables are constant, and always mention calculating a mean from repeated results to reduce anomalies. Avoid vague statements like light affects photosynthesis. Be specific. Increased light intensity increases the rate of photosynthesis. Always name at least 2 control variables and mention repeats and mean values for reliability. The method description must be a step-by-step process. Set up the pondweed, place the lamp, count and repeat the bubble count, move the lamp to subsequent distances, and calculate the mean. When explaining results, link the distance change to light intensity, state that light is a limiting factor, explain how this affects available energy, and finally, connect it to the rate and number of oxygen bubbles produced. Improvements can include using a colorimeter for more accurate oxygen measurement, a water bath for precise temperature control, and more distances to gather more reliable data points. A good conclusion has a 3-part structure. State the pattern observed, link the pattern to photosynthesis, and mention the role of limiting factors. When evaluating results, exclude anomalies before calculating the mean. Remember, repeatability means the same person gets similar results, and reproducibility means different people get similar results. Let's run through a quick-fire revision. The independent variable is the distance of the lamp from the pondweed. The dependent variable is the number of bubbles per minute. Control variables include temperature and carbon dioxide concentration. Why add sodium hydrogen carbonate? To ensure carbon dioxide is not a limiting factor. Why use a heat shield? To prevent the lamp's heat from affecting results. Why take repeat readings? To improve reliability and identify anomalies. Remember to take a minimum of 3 repeats and aim for 5 different distances for reliable data. Control variables must be clearly named and explained. The whole experiment sequence involves placing the pondweed, setting the lamp distance, counting bubbles, repeating at different distances, and finally, plotting the bubbles versus distance. You've got this. Remember your variables, that more bubbles means a faster rate, to repeat and find the mean, and the 3 limiting factors: light, carbon dioxide, and temperature.